DNA Depot welcomes you to Micropipetting Basics, Kit DD125. The following video will guide you through the steps needed to complete this experiment, Micropipetting Basics. In addition, it will give you hints, tips, and tricks to ensure quality results. Check your kit for the following items: red, yellow, and blue dyes, glycerol, alcohol, and buffer solution, micro-titer plates to mix the dyes, fixed volume micropipettes, micropipette tips, and finally, your pipette cards. In addition to these items, you will also need a pair of disposable gloves and a container to discard used pipette tips. To the first well of your micro-titer plate, place 10 microliters of red dye. Additionally, add 40 microliters of buffer. Be sure to discard the micropipette tip each time you reach for a new dye or buffer. Since each pipette tip holds 10 microliters, this will be 4 trips to the buffer solution with your pipette. With your 10 microliters of dye and 40 microliters of buffer, well number 1 will now contain 50 microliters of solution. For well number 2, we will begin with a clean micropipette tip and 10 microliters of red dye. Add to that 20 microliters of blue dye, again making sure you are using a clean pipette tip. Finally, finish off well number 2 with 20 microliters of buffer. As with well number 1, well number 2 should have a total of 50 microliters of solution. Use the pipetting chart on page 3 of your instructions to find the correct mixture for each well. Although there are 8 rows of wells on the plate, you will use only 7 of them. Lay out one of your pipette cards on a flat surface. With a clean pipette tip, extract 10 microliters of solution from well number 1 and place it on the first circle of row number 1 on your pipette card. Try to keep all of the solution in the circle. Repeat this step with the second and third circles in row number 1. Are you keeping the solution within the circles? With a clean pipette for each, move down the wells of solution and repeat this exercise. Again, we are only using 7 of the 8 wells possible, so you will have a blank row at the end of your card. In this experiment, students practice critical micropipetting skills. They prepare and deliver various dye mixtures to a pipette card. Each dye sample is diluted accurately and delivered in triplicate to the appropriate circles. When dye samples are diluted and delivered accurately, the 3 circles will have equal color and the dye samples will remain within the circles.